Thank you for joining me in this descent into rabbit hole. Trying to prove that there are alternatives to dynamic energy budget models, to the standard dynamic energy budget model, at least according to criteria set by Klanjšček in his 2020 paper. So here is a transport network model where food enters a reserve compartment and the energy, assimilated energy, divides up between the somatic and the reproductive branch. The somatic branch also is responsible for maintenance, and there is a energy reserve compartment on top, interacting with the transport network. Made a few assumptions. So we assume strong homeostasis, which really is necessary to get thermodynamics right. So all of the fluxes have the associated conversions, and dissipative fluxes are defined the same as in standard DEB. We also assume that there is that the volumes of energy reserves transport network scale with structure. Basically, your vascular network grows with you. That's the idea. And we'll assume that there's an area of interaction between the compartments that is proportional to volume, to some power, could be 2/3. But I argue it's, at least for vascular organisms, closer to 1, because vascular systems are fractal mostly, and the fractal scale area with volume. Next, we need to define the model. So this is derivation of the model. It's just stating the equations. The general idea is that a flux from one compartment to the other is proportional to the energy density in the one compartment times the area between the two compartments. So, for example, the exchange between the two types of energy reserves would be given by just the difference between the two fluxes. Let's specify all the fluxes. So these are familiar from the standard DEB. Assimilation, maintenance, let's ignore heating for simplicity. There is a somatic, flux to somatic branch is proportional to the energy density in the transport network and the area. Same for reproductive branch. The exchange is given by the previous equation, and the growth is just a difference between, is just given by what is left in the somatic branch once maintenance has been taken out. So these are the equations. Then just here if you want to scroll later, but we don't have time to go through them. So I've decided to cover just some of the stylized effects. There are 20 something and I have 15 minutes, so that's not going to work. These are the most dicey ones. G1 length of isomorphic organism in abundant food is a fully switching function of age. So let's just sum up the two energy equations. We get a linear closure term, which means that as ET grows up, it will eventually catch up with the V2/3, and ET scales with V2/3. Inserting that into the growth equation gives us here we get V2/3 and minus V. That's the standard literal for lateral entry growth equation. So we have maximum size or satiation function of H. So depending, just a full disclaimer, a total weight could start slowly reducing once the optimal size is reached. So the next dicey one is the scaling of the chromatographic growth rate. So inserting letters into the equation from the previous slide gives us this equation, and the ratio of the somatic investment into somatic branch versus total investment of energy is the equivalent of a kappa this model. And it compares actually favorably with equation 213 from the book for the growth. So both are, sorry, this is 213 for constant food. Otherwise, C is not a constant compare, but this is for constant food levels. And basically, it's the same equation. No wonder then that the L infinity is the same for both equations, and none of these have any reason to scale with anything. I mean, these are exactly the same, have exactly the same meaning in both models, so all the scaling should be equal to. Increasing weight of fetuses proportional to cube time. Let's look at the growth equation and note that ET here for fetus should be constant. So we have constant minus constant, which is a constant, times V, gives you exponential growth. And basically, that's the prediction of the model, except that ET, even mother's ET is not infinite. So exponential growth in a finite environment gives you something like logistic growth, here denoted by blue circles, and fitted by a cube function. It fits really nicely for this period of the fastest growth, but you have to move it a little bit to the right. And I think that movement to the right of the cube, what Klanjšček calls delay in ontogenetic development or something like that. But I think it's actually not a delay. It's just that the fetus is growing logistically rather than the cube. And but at any rate, the fit is good enough to proclaim this as, or to proclaim the stylus effect satisfied. But do please note the growth curve for the human fetus, which is actually logistic, not a cube. So I think this model actually, without additional parameters, does better in describing fetal growth than the standard DEB. Sorry, should have been skipped. Reproduction increases with size intraspecifically. Sure, that's the first term here. The bigger you are, the more energy you have, the more you'll be able to give to reproduction. If you're growing larger, BS is larger, BR is smaller, so you get less energy to reproduction. So I guess if you're a species that has the same assimilation but wants to grow larger, you'll reproduce less. So that works. I'm not sure, though, that's what Klanjšček meant by the second part of this stylized fact, because he didn't specify, and it's not specified in SOSA 2008 at all. So I'll put a question mark just in case here. So scaling the Kleiber law, DEB achieves this scaling by taking the somatic maintenance with a power of 1 and then reducing that power by surface dependent dissipation and energy reserves, whose fraction is increasing as animals get bigger. So the surface dependent dissipation, this model also has. Says now for the second one, it's not that clear. This is the quasi-equilibrium. Well, here it's actually exact equilibrium for the maximum size, and inserting LM into that gives us the maximum transport compartment energy density, and it doesn't obviously scale with length unless beta S does, or rather with size. And I think it's fair to assume that the rate of per cell uptake of energy from blood should go down with size. So I'll mark that as a success, but with a potential caveat. Moving on, now we're coming to the really interesting ones. The chemical composition of organism due to the strong homeostasis indeed depends on F, because V doesn't, right? The composition of V doesn't, the composition of E doesn't, but the amount of E does, and it's different from V, and you get a total change in organism composition. So that's okay. But at constant food density, does the actually the real question here is, does the weak homeostasis hold? And looking at this equation, you would say yes, but that's actually not true, because this is a quasi-equilibrium, so you don't have actual homeostasis. Now the real question, you have a semblance of homeostasis, but not a weak homeostasis, but not an actual one. Now the question, is that a problem? And I looked into evidence for weak homeostasis in Sosa et al., and it's not really convincing, at least not to me, because, so for example, Chiliard et al. paper, there really is no functional response here, just like one constituent and the other constituent, it's like flat and then jumps up. There are two time points in plasma leptin lipids body weight measurement, but that doesn't look like convergence to me. It's still changing. Plus, you can draw all sorts of curves through 2 points. Kroll et al. Has to feeding levels, but not confirmation and convergence. In fact, as time progresses, the composition keeps changing after 30 days, certainly after 70 days. I don't know whether now it converged or not because it didn't measure. And then the Engelbleek 2006 paper has only adults in there and has huge variance. So you can basically, well, draw all sorts of lines through this. The Fink et al. Paper has a variable food level. That's this graph, and here, sorry. And pretty much constant composition. So if you look, this is invertebrate C/N ratio. So even though the food of the invertebrates changes, the composition, as far as I'm concerned, stays pretty much the same. So it's certainly an argument for homeostasis, but not necessarily weak homeostasis. So I did not understand what's the point of the Tinbergen et al. References. So sorry, can't comment on that. So I'd say we're okay. We don't observe strict weak homeostasis, but it's good enough. Do note, though, that these approximate weak homeostasis is an emergent property of the model, only considered DEB, where it is assumed. So, and then dynamics derived from it. So we have some costs to this model, new state variables, new parameters, new stuff to think about, which nobody likes. Are there any benefits? So you get flexible reproduction. You can do demand, supply, whatever you want. You have simple addition of new processes. Just tack them up. They will naturally integrate into the dynamics. Multipurpose energy reserves. So you have to wonder where animals keep their reproductive buffer. Have independent possibility of independent parameterization, because with this additional compartment, you can actually measure a lot of these rates. And for whales, this is the growth curve we got back in 2006. With this model, none of the red dots were used in fitting. Even food was fitted from the was calculated from daytime reproduction. All the other parameters were done using purely physiological arguments. Similar here for Seabream. The fit is not as nice, but if you think that it could have been pretty much anything, right? So these years data like bone calorimetry being used in here, I think this is pretty close for a complete guesstimate of the growth curve. Not even, so we assumed food of F.8, maybe, but I mean, that's an assumption. I don't know, because it was in aquaculture, they tend to feed at 0.8. So anyway, The model gives us reason to treat smaller creatures differently. Remember the alpha. If you don't have a fractal cardiovascular system, maybe you use a different alpha and get different scalings. Reproductive senescence is built in. Different reserve turnover dynamics is may help with things like the isotope dynamics when well, don't have time to explain, don't have time for this graph, but the point is that reproduction and growth sometimes compete, and this graph illustrates that more quickly. So left is the formula amphi growth rate, fitting some fish data, and right is the formula growth rate with competition between growth and reproduction starting at maturity. The archaic information criterion of 20 gives us really a strong argument for using this competition-based model. And I think Laura had some anchovy experiences suggesting the same. So growth can respond to feeding and ignore reserve. This was a 2-month experiment. First, fish were differentially starved and then fed to the maximum. Even though they did not have the same conditioning, hence not the same energy reserves, they grew at the same rate. So that kind of that shouldn't be happening in a standard DEB. There are other stylized facts that are at odds with the standard DEB. So in standard DEB, toxicants distribute throughout the reserve or structure compartment immediately, thus acting as a low-pass filter for exposure. This is not the case in the transport DEB. So maybe the weak homeostasis does not hold. This is something I would like to see tested. Individuals of some species lose weight after reaching maximum size. We had a lecture on that by Ramon. Maybe it is due to lower assimilation, but it could just be the energy dynamics as presented by this model. That would, by the way, depend on ontogeny. So if reproduction is different, you may not observe that effect. And some species exhibit reproductive senescence. That's not, you have to attach a module to standard DEB, but it's kind of built in into the transport DEB. So there are some benefits in there. But I would also perhaps surprisingly argue that the transfer model is simpler because it's easier to derive. I'm sure even some of you now could derive it. So what you saw at the beginning was the whole derivation, right? And I don't know how many of us could derive the dynamics of the standard DEB, just some out on top of our heads. Easier derivation means easier understanding, easier modification, wider adoption. So I would, even though it has a few parameters more, and that's even debatable, I would call that, I would call this model simpler. But I wouldn't necessarily call it correct because it hasn't been tested. The standard DEB has been here for decades. We know that it works. We know when it works. We know when it doesn't. But that's not a reason to stop, right? We should look for alternatives, not maybe jump at them at any, without checking, but, and I think DEB theory in general is now mature enough that we can require tests specific to the theory. So how does homeostasis work? There are production, growth, compete. Let's test these things. Ask for funding that specifically lets us develop the theory further. Well, thanks to my financial overlords and to all the people that helped me and whose careers I probably ruined right now. And well, thanks. Okay.